And the classic nucleation theory, which there's other, nucleation theories out there, density fluctuations and the like, but to be honest, it's still one of the most practical descriptions of nucleation processes. And but typically, you'll break down the energy of the nucleation process into two separate terms. The initial term that is disfavorable is associated with interfacial tension. And then there's this other term which is associated with volume growth of the actual crystal. And so that interfacial energy term is disfavorable, and that's because so right here, as the radius of clusters of molecules as they come together and form larger and larger clusters, it's disfavored as they separate themselves from solution. There's an interfacial tension that occurs. But then once you get enough molecules together beyond that, the bulk growth becomes dominant and further growth, and the overall crystallization process becomes favorable and crystallization occurs. But some people say primary nucleation doesn't exist. I've heard that before from some pretty good scientists, but secondary nucleation, whether you consider it by seeds, which is something we use to our advantage in processes, lots of people, it'd be preferable that you wouldn't have to use seeds, but it's your best way to control crystallization of a process, and it's always prudent to write in a seeding step. You might not have to use it, but write in provisions for a seeding step in your final crystallizations for control purposes. But if you remove that interfacial tension through seeding, you can direct the crystallization to the form that you desire. And so if you have seed crystals, you bypass the disfavored process of nucleation. And so if you do the inverse of that, which is nucleation rate, basically the bigger the energy difference between the crystalline forms, the higher basically this nucleation activation energy. And so your metastable forms tend to nucleate at a faster rate than your stable crystalline forms. And so in the end, in the absence of seeds, when the energy difference between polymorphs are small, their nucleation rates are likely to be similar, and concomitant polymorphs more likely. In the absence of seeds, very large energy differences may result in very large differences in nucleation rates. And so the overall conclusions, this is kind of a reiteration of the earlier thing, but for the melatonin agonist, my inference is that there was basically a heterogeneous impurity, likely not the drug substance itself, because both of them were exposed to the same, the same environment. They were grown in the same labs with the same equipment. But it's likely that a chiral impurity enabled one enantiomer, heterogeneously nucleated one, whereas the other one was never exposed to that same mirror image chiral substance that caused heterogeneous nucleation to occur and crystallization of its stable form. But with that, I'd like to acknowledge my coworkers, Craig Wolfangle, Jennifer Runyon, and Dave Jackson for their analytical support, Professor Frank Lucien, who passed away, unfortunately, way too young, and John Kendrick, who worked with me on the ab initio calculations, which I didn't present today, but are in the paper. Thank you for your time and listening to my presentation. That's pharmaformfinders.com. Is the company that I work for as an independent consultant. And I thank very much Regis Chemicals for giving me the opportunity to speak today. can I get her first? She was up quicker.
So I just had a quick question, kind of on the last point you were making about there being a chiral impurity that you think was doing this. And so this is the chromatogram you have here. I don't know what's that. Is this the chiral method? Is there any?
You know what? You're right. This is not a chiral method.
Okay. No?
Oh, they would resolve.
Yeah.
Yeah. No, at the time we were doing it, but when I went back and visited, I did not have access to the resources, so I didn't have that data to include. Yes, I would have liked to have had that.
Yeah. Okay. That's interesting. Kind of the missing piece, right?
It is. Although it should have the same chromophore, right? But one of those in the same entities were present there. But yes, that would be worth knowing if one of those were chiral. I should have. Yeah, it's been a while. So, thank you.
I find it interesting that you said that a typical anhydrate versus hydrate, the anhydrate has a 4x better solubility than the anhydrate. Does that imply that the new polymorph is different? Are they interconvertible by a DVS, for example, with the curve? Or yes or no?
Yes.
They would be interconvertible?
So anhydrate, sometimes, it depends on the nucleation, how well it nucleates. So the question was, would anhydrate and hydrate if you're doing the vapor sorption, desorption, so changing the water activity in that piece of equipment. I find that if it's very soluble, the interconversion can oftentimes be absorbed be observed in the vapor sorption experiment. Sometimes I think if you really want to be safe, you're better off putting it into, because it's an expensive piece of equipment, and it takes a long time to make the measurement, or you're tying up a piece of equipment, it may take a while for it to nucleate the hydrate. 30 to 40% of the molecules will form a hydrated form. It's been shown at least in 3 or 4 good publications. And they always come up with about the same, the 30 to 40% of the time. So you should almost always expect that you're going to find a hydrate. You want to choose the form that is stable from, say, 30 to 70% relative humidity. It's nice if it doesn't interconvert. But to really be certain of whether or not a hydrate exists, DVS is extremely useful for understanding hygroscopicity, and whether or not it happens quickly changes from one form to another. But it's also nice to use, to place the sample over water at different water activities.
Right. What I'm getting at is sometimes you come up with let's say I did single crystal diffraction on the both, and it was essentially the same. It's just the water's there in one, not the other. Is that a different case than some hydrates where it's a different?
It is different.
So which is more typical?
I'm not going to say either of them is more typical. I've experienced probably half the time it's one where, and we oftentimes refer to those. So the question was, there's certain hydrates where the water molecule is an intimate part of the structure, and if you remove it, the structure will collapse. And there's other cases where you can remove the water by exposing it to low humidity or atmospheres of low water activity. And in those cases, usually there's a change in the lattice. It can be very small, but usually there's a change in the lattice, and usually there's actually fracture, so you can see it under microscopy. But sometimes the peaks stay almost exactly the same location, so you don't get a collapse of the structure. And those we call variable hydrates, and usually when we're doing our screening process, we try to stay away from those because it's more of a challenge analytically and other ways of controlling the potency, because you got to include water in the potency. So usually we try to stay away from those types, from selecting those as final products. But your cephalexin, which was called monohydrate, and then they made us change it to cephalexin hydrate because same thing with C-chlor, lots of molecules do that variable hydrate sort of behavior. It's just a little more challenging. Sometimes you have no choice, it's the only form you have, but other times, yeah. But I wouldn't say one's more common than the other. Run into them both.
A distinct difference in reversibility during TDS. Is that a difference between, say, adventitious water absorption? Is that a sign maybe you're seeing an anhydrate that has such a distinct physical property that it changes?
I'd say you're probably right. If you have a nice distinct step, usually that's a physical form change as opposed to like with cephalexin or it changes kind of, yeah, you get hysteresis, but it's not a real discrete change in the DVS.
The absence of that may not mean anything, but if you see it, it could be a strong signal.
Usually it's a good, yeah, that means it's a nice discrete change. The only thing is if you have hysteresis between them, it's a nice step, and you have hysteresis between them, you know that there's an interconversion, you just don't know where in that humidity range that occurs. But that's when we go on to slurries and solvents with water and determine what that critical water activity is to pin it down closer. So, thank you. Yes.
If you found a more stable form is because present related impurity, are you still going to pursue that more stable form or not? Or just
Yeah.
I need to present on this one.
Oh.
This amount.
So the question is, if you know that form is more stable, but you can't get it, are you going to market the metastable form because you can't get it? You know, I think that's what you would do if you had to, but I can tell you that if we had gone beyond phase 2 clinical trials, I can't imagine how many people in our company, we had a significant number of people working on it, but we would have had all the king's horses and all the king's men trying to find that polymorph. I promise you. Because somebody else could have patented that other form, and we couldn't make that form, so there's no way we could patent it.
In that situation, is it better to pursue the amorphous, like a square line?
Possibly.
Just to take that out of the equation?
That possibly.
It never really goes out of the equation, but you know what I mean?
You know, that I'm sure we would have considered that. Yes, it actually happened before people really went to doing more of those, but yeah, that would have been a good option.
So if you're young, phase 3, then with phase 3 study in the metastable form, and then you do the bridging study, show that because the solubility, you don't need a high solubility, maybe they are equivalent. Is that sufficient to justify that I don't need to repeat this phase 3
Yeah,
study?
you'll have to go through the ICH guidelines. Yes, you can go with metastable forms. There have been plenty of metastable forms that have been marketed, and then they've later found stable forms. But as long as they can continue to produce the same form, even if it's a metastable form, there's nothing that precludes you from developing that metastable form. But there's a risk.
Develop a metastable form. I said if you done with the pivotal study of metastable form, you found more stable form, but you already done with the pivotal study, and you just do a bridging study. Do you need to go back to repeat those pivotal study? You can say, well, that's crazy. So
I think that's good science, and that'd be dialogue between you and the FDA, or you and the regulatory body. But you could rationalize through modeling and other things whether or not.
So I have a question on your two enantiomers that you crystallized out. So did you have the single crystal for each of these enantiomers? So was there a conformation? So if you think about, there's an amide bond in there.
Yeah.
Right? So, and you have a different chiral center.
Uh-huh.
So you could potentially envision a situation where you have a chiral center, and then your amide bond, the way it sits, imparts sort of like an atropisomerism type thing.
Yeah.
You end up with two different diastereomers now. So they're no longer enantiomers, but what you have are the equivalent of an atrodiastereomer.
You
Is
could.
that possible? Or is that
I mean,
the
you
reason
could
why-
rationalize that, but that would be available to either one of them.
Maybe not, because that's where So in one of the enantiomers, the other, where potentially the other bond rotates and sits could be affected. That
Well, okay, yeah, you're saying, yeah,
so that
it's
would- that could
possible.
cause essentially like an atrial isomerism.
If it's a high enough energy barrier. I don't
Correct,
think in
between
this
the
case,
conversion of the two.
I don't think in this case you have a high enough energy barrier that, I mean, those are single bonds. I doubt it, but I read the paper, think about it, send me an
I
email.
was really curious.
It is, it's a unique,
I think
unique.
in your crystallization system, there must be some chiral effect. You didn't find, but other than that, there's no explanation why one form
Yeah.
from one or the other from two.
Yeah,
Maybe something there.
and I think that's,
Minor things.
polymorphism's unique and nucleation's still fascinating today as it was 100 years ago.
Yeah, and from your solubility, you're saying it's about 1.3, sorry, 0.8 kcal.
Yeah,
So
point, or fourfold.
conformationally, 0.8 kcal is not a whole heck of a lot.
No,
I mean,
no.
It's like that.
No,
that, right?
but I don't think that's your explanation, but anyway, if there's any other questions, if not, I'll let you guys eat.